So I just want to acknowledge that we're hosting this session on the unceded and ancestral territory of the Coast Salish peoples, including the territories of the Musqueam, Squamish, Tsleil-Waututh Nations, and the Métis Charter Community of the Lower Mainland region. And each of you is in your own region and territory. And I ask you to reflect on the value and the opportunities you get by being there. Next slide. So it really gives me, it's a great honor to be able to introduce Dr. Keown. So as many of you know, but not all of you, Dr. Keown has been a professor of medicine at the University of British Columbia and has had an amazing research career focusing on the immune response of transplantation and autoimmune diseases. He's been the executive director of the BC Transplant Program, the director of Immunology, and the head of the division of Nephrology. He's also been the president of the Canadian Transplant Society, the member of council and president of the International Congress of the Transplant Society, and he's a fellow at many international colleges and societies. But more than that, Paul is a wonderful researcher and also an outstanding educator. And I must say that whenever I hear a talk by Paul, I always learn a lot. And so I'm very much looking forward to precision medicine and epitope matching in kidney disease. So Paul, really with great pleasure, welcome to Province-Wide Rounds.
Thank you very much, Adira, for that kind introduction. I'll just move to share my screen and Brenda will tell me if it's working well. Brenda, is everything okay?
Perfect.
Yes. Everybody see. Thank you very much. So many thanks, Adira. We've been actually working in this field for quite some time. And what I would like to do today for everybody is to give you a sort of status report on our last big grant, which is a Genome Canada Award, which draws together most of the transplant teams across Canada and many of the teams in leading locations in other parts of the world, across the United States, across the European Union, and across the United Kingdom. And you'll see some of the various universities below. 22 universities across those regions with about 70 PIs. And today I would like to present it on behalf of not just myself, but James Lan, who, as many of you will know, has now taken over as the medical director of the Immunology Laboratory for the province. James, thank you very much in accepting that. And Karen Sherwood, who has been responsible for much of our technology transfer that I will describe to you, and is the scientific coordinator of the Genome program. So let me start, and for some reason we're not moving forward. Let me start with the problem. Transplantation is great. Canada is at the forefront of this field and we've made it a real success. So our early outcomes are superb. We have rapid recovery. We have patients going home literally in days post-transplant, returning to the regions around the province in a few weeks. Such a dramatic change from the early days when we labored to get the program going here. For children and for others, it now allows normal growth, development for adults, quick return to work, and it's enormously cost-saving by comparison with other modalities of care for chronic renal failure. But the problem is, as you see on the image on the right, that we achieve, let's say for argument's sake, an eGFR of 60, 70, or 80 within the first few months post-transplant. But gradually that begins to diminish. And by the time we become anxious and perform a biopsy, we often find acute or fixed changes which indicate long-term destructive deterioration and death. And what we hope to do is prevent that, because the consequences of this are really disastrous. Despite good long-term function, we have poor long-term survival, not just here, but really throughout the world. In fact, few grafts survive over 10 to 20 years. In Canada, more than 500 patients every year lose their graft, and the incremental costs to Canadian healthcare are staggering. On the bottom right image, you see a modeling program which Adrian Levy performed with us a couple of years ago. This is looking at the American data, but the Canadian data is very similar. And it says that even the patients with the best GFR, looking at their probability of functional survival, by 10 to 15 years, 50% of the grafts are lost. And those with poorer initial function lose them much more quickly. We know from a lot of work, but much of it's superb Canadian work, this from the University of Alberta, that the most damaging cause of graft loss and the one with the greatest potential for prevention is rejection. And antibody-mediated rejection is the particular target that we have focused on. And about 50% of the grafts that are lost to antibody-mediated rejection and a few to other forms of mixed probable rejection and so on. Overall, about 60% are lost to some form of rejection. But if we come back to antibody-mediated rejection, this is a fascinating response against foreign antigens on the target graft. And what happens is donor-specific antibodies are formed and they appear and disappear in waves over time. And they can cause acute injury or progressively chronic injury with gradual loss of the lumen of the vessels in the graft so that the graft dies an ischemic death. And both T and B cells are involved in a very complex interplay. And while the antibodies are probably the principal mediator, they're almost certainly not the only mediator. So in setting the program up, we drew on Canada's strengths, and Canada's greatest strength is that we work well together, and across the country, the Canadian Blood Services is the overarching umbrella organization with all of the programs that you see there. So we drew on these because as we applied to Genome Canada requires a societal perspective also. It says you may do fascinating work, but will patients agree to it? Will the public support it? And will people pay for it? And then how will you implement it across the country? So these programs are actually critical in diffusing the work that we are driving in British Columbia. And because this is such a challenging immunological phenomenon, we brought together teams working in different areas, certainly in our regulatory areas, because we envisage innovative interventions coming forward, scientific societies, genome laboratories. There are 13 HLA laboratories across the country which all participate, 22 transplant programs, people from pharmaceutical sciences here and in many other universities, and then law, ethics, and societal perspectives. And we asked 3 sets of questions. I'll focus really on number 1 today because I think it's the most interesting for us in British Columbia and the closest to fruition. So the first question was, can we minimize the risk of antibody-mediated rejection by improving the match between donor and recipient? This has been a real challenge for those of you who have been involved in the debates over the last 20 years. We've gone backwards and forwards and never been really able to achieve sophisticated matching, and I'll show you why. But we hope to be able to do that using the HLA genes that are the key drivers of rejection and matching on those in a new way. And then if we could do that, can we monitor the immune response more effectively, and can we personalize immunosuppression to support where we want to go? So I'll address this most deeply, and then I'll look at changes in these as well. So let me start with the whole question of immunology. And I'll start with the immune synapse. So when 2 cells come together, they can be self cells, an antigen-presenting cell and a lymphocyte, or they can be a recipient lymphocyte and a donor cell. They meet through what's called the immune synapse, or otherwise known as the supramolecular activation cluster. And if we imagine pushing 2 balls together, we get a flat plate between them. That's exactly what happens when these cells meet. You see responder cell, stimulator cell, you see the interface between them. This is called the SMAC, supramolecular activation cluster. And it has a series of concentric rings which are very carefully organized. So in the middle, we have HLA, or the MHC peptide, and the T cell receptor, which really transmit the key information we'll talk about today. But around them, they have a series of adhesion molecules that pull them together and also cause activation. And then on the outside are a series of regulatory molecules that move the system backwards and forwards. And some of these are interesting in themselves. CD43, for example, is the gene and the molecule that's abnormal in Wiskott-Aldrich syndrome. CD44 and CD45 are mutated in various kinds of malignancies. So these cells have many other, these molecules have many other functions in human disease. And at the end, I'll come back to us and talk about those. But today, I'd like to focus on this interaction here, assuming that everything else occurs as it's supposed to. So what is HLA and why do we care so much about it? So HLA was never invented to deal with transplantation, of course, until the time when we started giving blood transfusions or solid transplants. We never became aware of somebody else's HLA, except by sort of mimicry through infection with various microbes, which is a minor effect in sensitizing against HLA. But there are different kinds of HLA, so let me explain what there are. There are two major types of HLA. There are 11 genes in total, and we'll talk about 6 today. We call class 1, A, B, and C, class 2, DR, DQ, DP. And they live in humans in the major histocompatibility complex, which is on the 6th chromosome. And this is a very long stretch of very complex chromatin, about 4 million base pairs. It's the most gene dense in the human genome. And there are at least 200 genes living in this area. And in many ways, it's like walking into a strange forest. You recognize a couple of trees, and we recognize a couple here. But many of the other genes are going to be profoundly informative. And I'll come back to how we're going to approach those. So why do we have 2? Well, we have 2 because the MHC class 1 deals with intracellular peptides. So particularly viruses, perfect example, live in the cell, generate copies of virus, and then they're broken down through the proteasome. They're loaded into MHC class 1, taken to the surface, and it holds them out so that the T cell receptor can come by, recognize them, and destroy what is a virus factory. MHC class 2 is different. It deals with proteins which are abnormal outside the cell. So let's say a microbe is taken up into an endozyme, a coated pit into the endosome, broken down, then those peptides are loaded into MHC class 2. So distinct functions and distinct importance in transplantation. So now when we put a graft in, how do our own cells recognize the foreign antigens? Because our own cells have always been educated to recognize self. They work by looking at our own self-antigens presenting peptides. That's their only biological function. But when they're presented with a graft, now suddenly we have foreign cells. And the structure of foreign MHC is so different from our own that we recognize it immediately as a really nasty thing. And we can recognize it in 3 ways. First, our own T cell recognizes the foreign antigen presenting cell and recognizes the foreign MHC and any peptides in it. They can be anything. This is called the direct pathway. Or the indirect pathway, our own cells chew up the foreign cells, take all the peptides, and present them in our own MHC. Or now a new pathway called the semi-direct, where our own cells actually capture the whole MHC from the foreign cells and present them. So as we go forward, we have to think about all of these complexities. The ideal would be to match MHC, because we know if we have an HLA identical sibling, the outcome is great. We seldom have rejection. Even if we have rejection, it's mild. We almost never have antibody-mediated rejection, and the graft survives for 20, 30, 40, 50 years. But the problem is this. When I first came to BC in 1987 to start the immunology lab, we had no genomic understanding of HLA genes. We knew some of the antigens, but none of the genes. And over the past 30 years, we now have more than 23,000 alleles on these 11 genes. So huge number to try and match. In fact, it's so complex that we need a barcoding system to name them. So think of a family. This might be HLA, O2 might be Murphy. This might be Susan Murphy. But Susan may have a twin Sarah, so we have to distinguish between Susan and Sarah and so on. So it gets very complex. And it's really, really difficult to even think about matching at the allelic level, as we say. So what we'd like to do is think, is there a way that we can break this down, make it much simpler? And if we look into the MHC at the structural biology level, we have one helix, and the peptide that it holds would sit in the middle here. And now we know that much of this framework is the same between many of these alleles, but there are just a few little substitutions which make them different. And these are usually combinations of about 3 or so amino acids. Now we can identify all of these, and these are the points to which antibodies bind. They don't bind over the rest of the molecule, just to these few points. So if we could just match for these, epitope matching, it would theoretically make it much easier. And it's the same is true for both class 1 and class 2 molecules. So this is what we've done over the last couple of years. We've worked with teams internationally, and a person called Renal Duquesnoy, who was in Pittsburgh, has been the leader of this internationally. Renal is part of the team. To make a long story short, we have completed full sequencing of about 5,000 subjects using next generation sequencing. And using computational mathematics, we've translated these gene sequences to epitopes. Now, there are far fewer epitopes than there are alleles. In fact, what we can do is look at all the class 1 alleles we see on our waiting list here, and we can devolve them to just a few epitopes, and the same for class 2, just a few epitopes. So first, it reduces the complexity dramatically. We go from 23,000 alleles to about 160 epitopes, dramatic reduction. The second important thing is the way they're distributed in the population. Because if a marker is infrequent, it's hard to try and find people to put them together. But if a marker is frequent, it's easy to match. So we can look at the alleles. There are a few alleles here which are fairly common. 20%, 30% of the population might carry. But most of the alleles are rare. Only a few people on our waiting list may have many of these, and 5 at the most. And the same if we look at class 2 alleles. So while we could match these, it's really hard to match these. And bearing in mind that everybody has 11 genes, and 2 copies of each gene, that's 22 different alleles, this explains why we've never anywhere in the world been able to achieve allelic HLA match. But when we look at epitopes, surprisingly, they're much more commonly distributed. You can see 100%, some are in 95% of people, and almost all of them are common, whether we look at class 1 or class 2, which is really surprising. So we can begin to ask, why does this happen? Why are they shared? And they're shared because they actually are common clusters of amino acids that are seen across the gene products, HLA, B, C. They're shared across many of them. So we can see why the complexity is reduced. In class 2, they're not shared as frequently, but the numbers overall are much smaller. So we have only 59 alleles in the 16,000 HLA genes, and we have 91 in the 10,000 class 2 genes. So it makes it very, very much easier. Now we can ask, in our population, how closely would people be matched in BC? So bearing in mind that we don't actively match for HLA in BC, we first of all transplant children and the most urgent required, they're not most highly sensitized, and then we allocate on a modified next serve basis. So what does that do for us in degrees of matching? So this is a model we built, a simulated model to look at this using many of the sequence patients we had. And we asked how closely would people be matched, or rather, what would the mismatch score be? The maximum mismatch score is not quite 160, it's a little bit less, but let's say it's 100 for argument's sake. So the average mismatch score would be about 30. We have some who by chance are well matched, some who by chance are poorly matched. And we can look at the class 1, the class 2, and across each of the genes. So this is the prediction that our people that we transplant would be relatively poorly matched. We can actually test it because this data is from the time we started, 2018 to 2020. We can now look back on the preceding 10 years with real data, and we can say how close was our prediction to reality. And we can see it's exceedingly close. So what we predicted is almost spot on for what actually occurred. So the people that we transplanted over the last 10 or 11 years had in general poor matching because we didn't aim to match. So now the question is, if we tried to match with these populations, could we actually achieve it? So we can redo the model now, offering each kidney to the best match person on the waiting list. What would it do? And this is what it does. So it moves everything to the left, and particularly the class 2 genes. You can see the class 2 genes, many of them have 0 mismatch at VR, 0 mismatch, 0 mismatch, even at some here. as to 0 mismatch. So this is really encouraging because it says there's a real possibility that we could now achieve a completely new program of epitope-based matching in BC, which would arrive at 0 match. Okay, so that's encouraging by itself. What it means is we would take a curve like this, which is the probability of good matching, and this is the epitope mismatch number. Today, with no change, the probability 50% of patients would have a disparity of about 26, 27 epitope mismatches. But if we matched, we could actually reduce that to about 9. This is all 11 genes, about 9 mismatches. But within gene classes, we can see that for many of them, it falls to 1 or 0. And data coming from other sources, including Canada, so Chris Weeb and Peter Nickerson and others in Manitoba have been following this within their population, have proposed that an epitope match at DQ or DR, which is less than 10, gives us a very good outcome. And we're actually saying we could maybe get 0, which is really super. But the question relates to the size of the waiting list. So if we have a small waiting list, it will be difficult to match. If we have a big waiting list, it will be easy to match. And the key question for us in logistical terms is, do we need to share organs all across Canada as we do with a highly sensitized program in order to get the size of waiting list we need to match? If so, we can do it, but it's an expensive and challenging logistical problem, and it increases cold time. So what we've done here is we've modeled all of the waiting lists across Canada and then Canada as a total. So Saskatchewan has the smallest wait list with about 80 patients. BC's in here. BC has about, we've modeled 250 active patients because there's a lot of patients on everybody's list who are on hold. So we've been very conservative, and Canada has about 2,000. So what it says is for each of the genes, waiting list size is important, and the closer you get to perfect matching, the more the waiting list size is important. But in principle, once you get to about a waiting list size, this cut here is about 250. Once you get to a waiting list size of about 250 active patients, you have a really good chance of excellent matching. And these show a match of less than 10 at DQ, DP, DR, and here, again, DQ beta 1, DQ, DR beta, all of the key genes which really drive the immune response, the class 2 genes, we have a wonderful chance of even a 0, let's say an over 80% chance of a 0 mismatch, even just allocating within province. So if we looked at this across Canada, BC could probably do everything by itself, Alberta could probably do everything by itself, Saskatchewan and Manitoba might pool, Ontario could do everything by itself, Quebec and the Maritimes would pool. There's a little wiggle in the thing here, and it's BC that causes this problem. Because as we entered into this, we saw it's not just the waiting pool size, it's actually the number of donors who are available. So the more donors, proportionately, the smaller waiting list you have available. And this is BC, which disturbs this because we don't have a very big waiting list, but we have a large number of donors. So we would normally be up here on the rest of the panel if we took the same numbers as everybody else. Now, we talk, this reflects direct recognition. This is immediate recognition, formation of antibodies which come back and bind to those targets. But there's a second approach called PERCH. And this looks at the indirect presentation of antibodies. It says, okay, our monocytes, macrophages, dendritic cells will go to the graft, chew off a little bit of the graft, digest the HLA antigens, turn them into peptides, and present them by themselves to our own T cells. Can we calculate this? So this is a different computation because each peptide that fits into the HLA molecule is about 9 amino acids long. So a long amino acid with 300 or 400, the 350 amino acids in it can be broken down into thousands of different stretches of 9 amino acids. So we have gone through this computation. And this data I'll show you here comes from our sister group in Berlin, who we're working with also, who have put together the matching we have looked at, the one I've just shown you, the B cell matching and the T cell matching. And they look very similar. In other words, if you match well for one, you usually match well for the other. It's interesting, though, they're not identical. So some people are up here matching well for the indirect presentation, but poor down here for the direct presentation, and vice versa. So we're recalculating now to look at all of these implications for BC. What does it mean in practical terms? Perch score is a little bit different. And so they break it down into regions, less than 9, 9 to 35, 35 to 90, and so on. And the development of donor-specific antibodies is related to those scores. So what we try to do is make sure we get a score as low as possible, but in practice, it will depend on how well we can match. And here, this is the figure I showed you for the B-cell epitope matching. We can do very well. This is the figure for perch because it's more complex. And what it says is that we can reduce it probably to below the 35 threshold for most patients. Now, we don't know whether this or this is the most important driver because occasionally they go hand in hand and occasionally they're separate. So we're using our retrospective data from British Columbia, looking at the outcomes, trying to answer this question, then we'll know which we want to match for. So this is great. We can match, we can type the recipient, we can get their epitopes, we can put them on the waiting list, we know what kind of donor we want. When the donor comes, we can put the algorithms into the computer system so that it will immediately tell us who to pick. The only thing we've not been able to do is to identify the epitopes of the donor quickly. We only have a short period of time, and we've set ourselves a period of 6 hours for argument's sake, because we want to get every kidney in within 12 hours if we can. And the way we define the epitopes for the recipients, where time is not a problem, is by next generation sequencing. So BC really led the field in next generation sequencing, and we're now beginning to apply it all across Canada. And it's done by an Illumina method. And this method takes very short reads of DNA, and like building a wall with bricks, it puts bricks spaced between each other, one on top of them, until you've got a big depth of bricks. And then you can look at it and you can say, yes, this is HLA 0202, or whatever it is, doesn't matter. So it's a long process that takes us on average about 4 days to sequence. So clearly no use for donors. But there's an exciting new method called long read or nanopore sequencing. And we talked a little bit about this as we were thinking of bringing it on. And thanks to Karen and James, this has moved really quickly. So in the space of less than a year, we've gone from very first steps in this field to now having validated it and it being almost ready for clinical use to replace our old methods. And what it does is it takes a strand of DNA, a long strand, and it pulls it through a pore, and as each base comes through, it reads the base, and there are only 4 bases. So then it gives us the immediate sequence. So we can see the sequence according to the different base here. But the beauty about this is the sequencer itself is so small. This is the sequencer, that's a laptop, and we put the patient on, and it will read it within a period of 6 hours. So less than 5 hours for everything from starting to prepare the sample, to running the sequence, to doing the calculation. So this looks as though it will be the method of choice for sequencing for donors. Because ideally we would like to have similar methods for donors and patients so that we don't miss important information between them. But not every lab is going to be able to do this. So we need simpler tools for smaller labs. Unfortunately, there are smaller tools. There's a little kit which is now available, which is a real-time PCR kit, runs on a little machine like this, which doesn't give us the level of sophistication we need, but we can use computational methods now to enhance it. And it looks as though we can get 100% prediction accuracy of the epitopes we need within 2 hours. So even the simplest lab, the smallest lab, should be able to do epitope typing of donors and of recipients. So now we come to the point where we have virtually everything in place. And we're beginning to talk now about the possibility of launching a proof of concept trial of epitope matching in British Columbia. So we have all of our recipients sequenced, and we have all of their epitopes. We have the methods now for typing every donor. In fact, we do this routinely just for practice for every donor that comes along, and we can begin to select. So we're talking to all of the groups now, John, Jag, James, and others, and BCT, about what are the steps we need to take? How do we inform the physicians across the province? How do we inform the patients? How do we calculate some sort of benefit versus detriment? What does it mean? So let's assume we have 250 people on the waiting list. At the moment, Mr. Brown, who is number 2, will get the second kidney that comes along. And Mrs. Jones, who is 10th, will likely get the 10th kidney that will come along. But under an epitope matching scheme, that would be changed. So Mr. Brown might have to wait until his good match comes, and it might put him back to 59th, and Mrs. Jones might go to 110. Whereas somebody who's just come on the list today could be the perfect epitope match for the next donor and would lead right. So we have to deal with this question of selection really importantly. We want to be transparent, and we want everybody, particularly the potential recipients, whose time will be altered, to understand. So we thought what we can do is we can portray it to them in a way that says, we have a great opportunity here to increase success. If we can increase success, it means you keep your kidney for longer, potentially for your whole lifetime. The potential drawback is your time on the waiting list is no longer so clear. If you were number 10, we could say you'll get your kidney in a few months. Now we can't say that. But what we could say is that if you have not received a well-matched kidney by a certain time, we could say 1 year or 2 years, which is the sort of average waiting time that we're looking at the moment, then we will immediately give you the next kidney that comes along. So your waiting time would not be longer than for any person on today's list. So those are the kind of discussions that we're thinking about, and we very much appreciate your feedback because we want this to be a provincial undertaking. So then let's assume we can launch an epitope matching program. It would be the first globally, and it would clearly be a leader in its field, and we think it would be very successful. There are a few additional things we want to do. We want to improve the ability to monitor the immune response, because our ability at the moment is very poor. We measure antibodies, but almost nothing else. We know that uremia is a profoundly immunodisordered state, and we have 2 things going on at the same time. We have enhanced inflammation with impaired allorecognition, recognition of microbes, response to microbes, and so on. And we're beginning to try and tease these apart and understanding why they're abnormal and what happens over time. How do they improve? Do they return to normal? And then we want to find a very specific measure of the response to the graft. So if I get a graft from Mrs. Smith, I want to have a flag which says very early on, before antibodies are ever formed, yes, you're beginning to respond against Mrs. Smith's graft. Now you need to adjust your immunosuppression. So in pillars 2 and pillars 3 of the program, this is exactly what we're trying to do. And we're following in a biolibrary, we're taking baseline samples, samples from patients during quiescent periods after the transplant, samples from patients during rejection, and we're looking at global immune competence, we're looking at donor-specific immunity, we're looking at antibody responses, and we're looking at measures, subtle measures of graft injury before biopsy. And we're using a series of tools which are literally just evolving almost as we speak. These are so new that if we'd been looking in the literature 3 or 4 years ago, we wouldn't have seen any of them. So we can do deep immunophenotyping. We can take cells now and we can look at 20, 30, ultimately 50 or 60 markers on their surface that give us exquisite subtypes of these cells and include activation antigens. So not just is this cell a specific type, but it's actually in this stage of activation. And then we can look inside the cell to see what the protein signaling is doing. We've talked about nanopore sequencing. We've used it for HLA, but I'm going to describe how we use it for other things in a moment. We can use microfluidic proteomic analysis. So antibodies are a real problem for us. I'll give you an overview in a moment that shows, yes, they're really important, but they're very, very cloudy. There are some people with antibodies who have no problem, people who have what appears to be antibody-mediated rejection with no antibodies. And James has a big program now looking at the specific targets of these putative antibodies to see what they are. And then all of this comes together in what we call single cell biology. So at the moment, we take 1 million cells, we put them in a tube, we look at different functions of them, but we can't get the function of each cell individually. But this will allow us to. So as each cell moves through a staging system, we're able to define what type it is, its phenotyping. We're able to define its genetic sequence and its genomic transcription. We're able to look at the proteomic signaling, then put it all together for every single cell and rebuild the picture. So this is just one of them. This is flow cytometry. All of you know the principles of flow cytometry. We flow a cell through a fluidic sheath and we shine lasers on it. It has antibodies on the outside. And we can quickly distinguish lymphocytes, monocytes, granulocytes. We can sort of work on the subtype. But this is what we get. We get pictures that look like this, and then we get sheets with hundreds of numbers on them. I mean, James and I work with them every day, and it clouds your brain and you go dense after no time. You cannot hold this information in your mind anymore. So what we're doing now is we're using machine learning and artificial intelligence to create pictures. So what we do is take all of this data and translate it into a picture like this and a sequence like this, where each node, it defines a specific cell subtype, a very fine cell subtype, and the color defines qualities of those and numbers of those. And you can immediately see the transition. So these cells go from here and then they morph into these cells. And if they take this path, they morph into these, or this, they morph into the other ones. So suddenly you can get functional and structural information from the same. We're looking at genomics, and previously we've done a lot of work looking at how cells recover, how they're sick in uremia, and they're profoundly sick, and how they recover. And this is a person post-transplant, and you can see some genes, each line is an individual gene over time, remain profoundly depressed, 90% suppression of this gene, whereas others return towards normal. But it's hard to tell in which cells. What we do know is if red here is normal, if this is the gene sequence across all of these genes for all of us, the transcription rate rather for all of us, and this is the patients in uremia, each gene, each line is a different gene, we can see overall they're profoundly depressed. And these are patients doing well post-transplant, and you can see they're better, but they're not back to normal. So even though the patients that we all see every day look really well on a genetic landscape, they're not yet healthy. So what we want to do now is to be able to use single cell genome sequencing and take the blood from these patients and run it for each cell and see what the profile is in each cell. And then understand, yes, this group of cells are healthy, but this group of cells are sick. Why are they sick? And we know there are some master genes that control hundreds of others. And maybe if we can influence the function of the master gene, we can actually return these to full health here. So now we can take this a little bit further, and we can actually say, okay, we know what HLA presents. It holds the peptide, and the T cell receptor recognizes it. And science tells us that each T cell has a unique receptor. So each T cell is a clone with a uniform receptor. And when it recognizes a very specific antigen on the donor, that clone proliferates, divides. So theoretically, the number of copies of that receptor should increase because there's one, at least one, in each cell. So the more receptors, the more cells in that clone. So this was started by Megan Sykes, who was then at Boston, is now in New York, where she took a mixed lymphocyte culture. She'd take cells from subject A, cells from subject B, put them together, let them proliferate because they do in the test tube, take the cells, sort them out, and then look for the numbers of cells with unique receptors by sequencing the receptor. And these are examples of the profound numbers of receptors. There's thousands of receptors. But briefly, what you saw is in the bone marrows, which become tolerant after about 6 months, the numbers of receptors responding to donor and the proliferation is dramatically reduced by comparison with those that don't. So it gave us a door, an entree into this whole field of using T cell receptors. And we have followed that on. So we've been doing this in kidneys now. And we can see that as we do this culture over several days, from day 1, 3, 4, 5, 6, 7, the cell population changes and the stimulator cells die off and the responder cells, let's say this is donor and this is recipient, responder cells proliferate, some more than others. Now we can take these cells and using the nanopore sequencing, which I showed you, we can begin to look at the clones which evolve. So here, let's assume each different color is a different clone. We have lots of resting clones, but when we're stimulated, suddenly clones begin to develop. And we believe those are the clones that recognize the donor. So in fact, here we see clones which recognize the donor, which are dramatically different from the baseline. So what we're going to see now is if we can use this as a very early index of the first step of recognition in the immune response to the donor. So let me just draw to a close with some of the common things. One of the major problems is antibody testing. We do antibody testing all the time. James is just organizing a standard approach to antibody testing now, which is hopefully going to be approved by the hospital very soon. And this will give us a guide to the outcome. But we have to bear in mind that this is still only a rough guide. This is a wonderful systematic review by Jeremy Chapman and team from Westmead in Sydney. And what they say is, overall, these are all of the different articles they've selected out of 2,300 articles that reduced it to 36 that were informative. But you can see the heterogeneity. So there is an increased risk of cellular rejection and of antibody-mediated rejection, but maybe a little bit of overall graft loss and some mortality. And when they look, they start with 2,300 articles reduced to 36. When they look at each of the questions, the level of evidence which currently exists globally is low, moderate, very low, low, very low. So there's a huge amount of information to still be derived from this. We want to find ways of detecting injury to the graft before it happens. And one of the ways is to look for donor circulating DNA. So as the donor tissue of the kidney or the heart or anything is injured, it releases DNA. Now we can sequence circulating DNA. We can find our own and we can find the donor and we can quantitate the two of them. So very quickly we have a biomarker for looking at very, very subtle degrees of injury in the target tissue. And we're working in collaboration with groups across the world, but particularly in Europe, looking to implement this here very soon. So that brings me to close on systems pharmacology, where we have just started. So we have a wonderful database in British Columbia, thanks to PROMIS and the reporting that everyone's done over the last period. We've taken a period from 2008 to 2018, because if you like, that's the era of modern transplantation. That's when we've used antibody testing, we've used selection procedures, where we've used common immunosuppression. So what we're suggesting we can do is we may be able to define higher risk of AMR, lower risk of AMR. Those at higher risk would be those with a high immune response, who metabolize their drugs very quickly, and who have other problems and maybe some non-adherence. Those who are at low risk of infection would be those with lower immune response. So these might be children at the top, these might be older adults at the bottom, and then they have low immune response, they metabolize their drugs very slowly, and so they would need less. And in fact, if we give them too much, then they become over-immunosuppressed and develop malignancy. So one of the questions we're looking at now is, can we define what induction strategy we really need? At the moment, we use a rule of thumb. If they're highly sensitized, we give them ATG, but that's probably completely untrue. And we hope very soon that we'll be able to show that's the case. Then we want to see, how can we safely reduce immunosuppression, particularly in the elderly or in quasi-tolerant, operationally tolerant people? And how can we carefully enhance immunosuppression in those who are at highest risk of graft rejection? In going through this data, we're really just getting it ready now, but what we do see is that the success in BC is actually very much better than we had anticipated. These are our survivals. This is patient survival is good. The elderly die more frequently than the young, of course, but when we look at graft survival, it's exactly the opposite. It's the young who lose their graft more frequently than the elderly. This is death censored graft survival. So clearly we're beginning to see where we need to focus and what we need to do. James has done some wonderful work looking at the dosing and continuation of immunosuppression, showing that over the first year, a dramatic number, 50%, have a reduction in their immunosuppression dose. This is just MMF, but he's doing the same now with TAC and others. The reasons for this are leukopenia. May not just be MMF, may be viral disease as well. And so we're layering over this now viremia to see if there's a parallel. And the consequences of this is if you have your dose reduced for any reason, your probability of survival is reduced. So let me close now by just saying this has been a wonderful, wonderful voyage for us for the last couple of years. We've got a tremendous amount done and a lot more coming, and we'll be very happy soon to bring it forward to you. But it opens another new portal for the next generation. So we started by talking about the immune synapse, where the two cells come together and exchange information. That immune synapse is critical in 4 forms of immune disease. In transplant immunity, of course, as we just discussed, but also in recognition of self and altered self in autoimmunity, in infectious immunity. So what we have done, I won't show, is to use exactly the same approaches now to predict susceptibility to all of the key viruses that afflict the transplant patients, including COVID. And we can now identify who are the people who are at vastly enhanced risk of the virus versus those who are dramatically reduced. And then the 4th one is tumor immunity, the recognition of cryptoantigens or neoantigens on the target tumor tissue occurs in exactly the same way. So we think that the future will in fact not be limited to transplantation, but will be a concept of immune sciences that begins to link all of these in the same way, using the same information and the same tools. So let me close with my thoughts. We've come a huge way in a short time, and we're enormously indebted to everybody. I think we're on the verge of really being able to propose to you and to everybody a proof of principle epitope-based matching program that would start in BC and move across Canada. And very shortly, we will have the supporting mechanisms for monitoring the immune response, the studies which are required, and a variety of other tools to really help us understand what's happening post-transplant in a more sophisticated way. So thank you, Adira. I'll hand back to you.
Thank you, Paul. As always, exciting and informative, and certainly the whole area of immune health, as you've called it, is something that we're all looking forward to, especially in the current era of the world that we find ourselves in, with all the uncertainty of who does and doesn't get sick when exposed to the current virus, let alone if you're immune suppressed. So really terrific. I guess one of the things, how close are we, do you think, to being able, in the first instance with the epitope matching, to from the get-go modify our immune induction therapy and subsequent therapy? Because, you know, certainly that at the moment seems to be a one-size-fits-all, irrespective, with a very crude thing. So I'm assuming or wondering whether or not with this better epitope matching, one could then customize how one looks at induction and maintenance therapy.
Yeah.
Is that correct?
The answer is absolutely yes. And the question is, how do we get to it? So our heuristic at the moment, our rule of thumb says if a person is highly sensitized, they've had a previous transplant, then high risk, we should give them epitope. But it's probably untrue. And the group we think will tell us that is the Canadian highly sensitized program, where we're transplanting people with 99% antibodies. So we're just discussing now putting forward a proposal to the CIHR to map the structural biology, the epitopes of those people, and the outcomes. And if they do well, as we think they will, as we think they have done, then it will mean that we can completely relook at the use of ATG and other tools. So you're right, Adira, we don't have the answer today, but I think, you know, within a year in BC, we would have that answer.
Great. So I remind the audience that in the Q&A, if you can write any questions that you may have. I guess I'll just, while I'm waiting for questions to come in, you alluded to the fact that we have to communicate with patients this potential change. And I know I've been a part of some of the participatory genome conversations that we're having in terms of how do you explain to people that they'll still get a kidney, but it might be a little bit later, but it'll be better, but don't worry. I mean, it's a kind of an interesting concept when people have been holding on day in and day out. So what are the challenges you're finding with that?
So there's no easy solution. You know, you and I have talked about this, and we've chatted with John and everybody across the country. I think probably there are 2 periods. There's a period while we have a washout of the current people on the list, because they have an expectation that they will be next up for that. And then after that, everybody will come into a new room. So how do we deal with 250 awaiting? I think we have to say to them, honestly, we think it will give you a better outcome and really the potential for long-term survival. But we can perhaps guarantee that you will not wait longer than, and then we can set the time. So we could say 6 months, we could say a year, we could, whatever we want. Shouldn't be longer than 2 years, I think. But if we do that, we can give them an understanding that yes, I'll participate and I may do better, and I won't have to wait too long.
Yeah, so there's a crossover period and you're imagining it to be a couple of years all in, right?
That's right.
Yeah. So there's a question in the Q&A. Can we use CRISPR technology to alter HLA genotype and help with better matching?
Yeah, fabulous question. So we're working with a group at the University of Madrid who are doing exactly this now. They've been using a variety of approaches to eliminate the HLA, the disparate HLA antigens on a graft. And so there are HLAs which have normal genes, but they code for a null antigen. In other words, the protein isn't produced. And all you have to do is substitute that mutation with CRISPR into the gene, and it produces no antigen. So you would have a kidney which has no HLA antigen. So we've talked about this. The problem is you probably don't have time to do it with
living donors, right?
But actually, we've been talking with a group at UBC for their islet cell program and saying this would be a wonderful approach in islet cells, right, which then you have a standard batch of islet cells which have no HLA on, you can use it for. And the group in Harvard I've just been corresponding with are actually looking at doing this in a formal way. So I think a lot of people are beginning.
Yeah, no, certainly lots of excitement. I'm quite conscious of the time. Any other questions? I mean, I guess the other thing is how much of what we've talked about, or I can imagine, but in terms of applying this same sort of precision to therapy for autoimmune diseases. It's a bit trickier, I would imagine, but perhaps some interesting offsets and corollaries that one could think of.
Absolutely, in two ways. So we're working with a group in McGill who have come from a background in autoimmune disease, and they are doing this T-cell receptor sequencing in diseases like diabetes and others, and now they're applying it to transplantation. So we can see crossing the bridge in the other direction will work very well. And the other is autoimmune diseases like psoriasis have become the test diseases now for all of the new biologics because they're so easy, safe, and we're going to find a wonderful family of biologics that could then cross the bridge into transplantation. So I think having the two together is enough.
Yeah, truly a fantastic time, I think, to be in medicine as well as in this field. So thank you very much for all the work you've done and are doing, and the teams that you work with. And it really is wonderful to see the Canadian and international collaboration. And thanks everyone for joining. We had at peak about 46 people, which is pretty terrific.
Oh, that's great.
So thank you very much, Paul, and thanks everybody for being here. Have a great rest of your Friday.
Thank you. Thanks to everybody. Thanks again. Bye-bye.